Hello.
Hello, how are you? Oh, hi, Tom. How are you?
Hey, everybody.
Hi. Hello, Jesse and Akshay. Hello, Tom. Yeah, I haven't seen you in a while. Wait for a few more people. Okay, I don't think people heard me before.
No.
Okay. Hello, Tom and Dick and Jesse and Akshay.
Hi.
Yeah. How was everyone doing?
Doing pretty good. I just finished teaching, so done with that.
Okay, that's great. Yeah.
Yeah. Well, this is finals week. I got a great stuff up.
Right. Yeah.
Now I'm done.
What are you teaching?
Just general computer stuff over at Kishwaukee College. It's a community college.
Yeah. Yeah.
Yeah.
So it's just like an intro course, or like?
Well, mostly, yeah. See, like, actually teaching JavaScript, which I don't know much about. I learned a lot, though.
Yeah. Way to go. Just.
Yeah.
Anyways. Well, I'm glad to see you here again, and.
Yeah, I'm glad to be back, yeah.
All right.
I'm going to see if I can get a camera on this other machine.
Yeah.
This one doesn't. So I might dip out and come back in this time.
Okay. Hello, Tharun. Got one. Oh, hi. Yeah. Okay.
Yeah, hello everybody. It's it's a very busy week for me on many fronts. I have like a bunch of stuff going on today that's going to decide probably what will happen in the next few months of my life. So I'm occupied with all of that, and it's all kind of like, I don't have much to say right now, but I think next time that I'm in a meeting, I'll have interesting updates. And I'm really excited to see what happens with the summer code here today as well. So
Yeah.
welcome everybody.
Yeah. So this is, Summer of Code. They'll be announcing Summer of Code this afternoon. So good luck, everyone. Anyways, yeah. Let's see. So I'm going to start with, I'll start with some information here. Let's see, share my screen. Okay, can everyone see my screen? Okay.
Yes.
All right. So first thing I wanted to mention was because our the GSOC results are being announced, we're heading into the community period, and so like I said, even if you don't get selected, I would welcome you to participate in some other way in the group, and this will lead to, this might lead to other things down the road, so don't feel discouraged. But I also wanted to point out to something that we have and we'll be continuing to build. This is the onboarding guide, and I talked about this before, and this was really built originally around the GSOC projects, but now that that application period is done, I've kind of adapted it just as a general onboarding guide. And so if you've not seen it before, and a couple of you haven't, this is a guide that we're using to get people into DevoWorm and sort of get them used to the community. So I think a lot of you have been at the meetings, already know kind of what's going on. But this is something maybe for more casual participants or people who might want to join but don't know what to expect. So there are all sorts of resources we want to build into this like, links to model organism biology and biological data sets. So those, once those are in place, we can do, we can basically onboard someone pretty quickly into the group. And then, if we have next year's GSOX projects, will be listed in this onboarding guide. And, we might even add in like potential projects where, kind of following up on the things that, from this year's GSOC projects, what can you do is sort of like a good first step into the community. And I think some of the some of the GSOC projects were sort of that. They were allowed people to do some quick participation, some quick commits, and get embedded into the community. So hello Yash and hello Aswarth. So that's the idea behind this, and anyone's welcome to contribute to the onboarding guide here. Did anyone have any thoughts about that? Okay. Let's see. I was wondering if people had anything they wanted to present before I move on, so we can give people time to do that. Okay. I just wanted to check. If you do have something, let me know, but I'm going to keep going. Okay. So I think next what we'll do is we'll talk about the submissions documents. So the submissions, again, this is our document for submissions. This is stuff that we've submitted or going to submit that's been accepted or rejected. So we're just keeping a running tab of things. We've had a couple of these talks on DevoLearn, and those have been completed. So we presented at the INCF Neuroinformatics Assembly and the OSF Virtual Conference. And we're still, this preprint is still kind of hanging out there. I haven't had time to get back to it. But there are a couple of opportunities that have come up in the last couple weeks, and I don't know if I mentioned... Well, I mentioned the NetNeuro and the TopoNets submissions before. So that's 25 and 5. And they got accepted this week. So the abstracts got accepted to the satellite session. So this is the satellite of Networks 2021. So we have a full submission that got accepted in the main track at Networks 2021. And this is the website for Networks. As you'll remember, this is a conference on network science, and we're in the mix there with a 15-minute talk in the main session. But there are these two other sessions that there are also presentations. Actually, the NetNeuro is actually a poster, but that's okay. It was a pretty competitive venue. So this is the NetNeuro 2021 satellite. So this is going on over several days. And it's a pretty big group of people. I think it's one of the bigger satellites of the conference. And so this is a lot of people doing stuff with like neuronal networks instead of neural networks. So these are brain networks where people are looking at connectivity in the brain. And they're kind of figuring out what's connected to what during behavior or maybe just anatomically. And so I know that's a little bit not really what we're doing in this group. That's why we got a poster accepted. But basically, there's going to be a lot of information about this area. So there are going to be a lot of big names in that area speaking. They'll be talking about the C. elegans connectome. But they'll also be talking about human brains. They'll be talking about other animal brains. And I'm sure it'll be a good time. So this is the abstract that was submitted to the NetNeuro. And this is the... Okay. This is the abstract. So this is Neuromorphogenetic Patterns in the Theory of Deep Learning. So this is a rather short abstract with a lot of references. And this was contributed by a number of people in the group. Mayuk, Mainak, Krishna, and Jesse have all kind of commented on it and shaped it a bit. And this is this is going to be turned into a poster. So the poster presentations work like this. You have a person presenting in a room, and they put a poster up in a large square. It's like a single slide or a single PowerPoint slide, and then you present it to people in that room. So it's like a breakout Zoom room, if you know what those are. And you present your poster at the allotted time. And so for something like this, a poster provides both benefits and drawbacks. The drawback is that you can't write out your ideas and arguments, but at the same time, a poster allows you to sort of visualize your ideas and create visually compelling displays of what your idea is all about. And so I think this will make for a pretty decent poster. I think we can work it out. I think this will work out. So if you're interested in participating in this, please let me know. I think actually we're open to contributors. So the way I usually do this is that if you contribute a reasonable amount, you get authorship, and you get an opportunity to be a part of something like this. So that's for NetNeuro. And then for TopoNets, this is the TopoNets site.
Do you know when the NetNeuro updates are due by? The poster?
I think the poster is just going to be kind of like up to the time of the workshop or the satellite. So I don't know the absolute dates on that, but I think it's going to be pretty close to the deadline here. So it'll be in June sometime. Yeah. I'll be circulating a draft as I usually do for these things. So, and I'll just put it in the Slack or I'll mention it in the email. So that's that. And then there's this TopoNets. So this is a different satellite session. And this is, so the aim of this satellite is to look at across like biological, social, and technological systems and look at the richness of interaction among their units. And so the way network science usually works is they take a bunch of nodes representing objects and they try to figure out the relationship between them. So it could be like pairwise relationships, or it could be multiple, one-to-many relationships, things like that. And so you end up with this network topology based on nodes and then edges which connect the nodes. But they're interested in this workshop and they call it TopoNets because they're interested in things that are not nodes. So they're looking at higher order architectures of complex systems. So if you take a typical network and you decompose it into some higher order structure, that's one example. There are other examples from what we've talked about topological data analysis, where you have shapes and you try to characterize shapes in terms of mathematics. So that's what this workshop is about. And they found it compelling enough, the submission. This is the Euler Cycles for Life submission that I presented in the group. They found that compelling enough to have in the workshop here in session 1. So this is that?
Bradley, could you give an explicit example of relationship between networks and these higher order phenomena?
Well, yeah, so networks show sort of like the connectivity between a bunch of elements in a network. So higher order network structures would be like taking like if you had a bunch of nodes and they were connected by edges, trying to figure out sort of like the global signal across the network. So people usually look at like motifs, which are like maybe a couple nodes connected, or they look at like the, maybe very broad statistical signatures. And with TopoNets, they're trying to figure out sort of like what the structure means. So like the shape of the network, or like, if you have spaces in between the nodes, maybe there's some spatial significance of how the network is structured. And you're looking within those, between the nodes for like, signatures in terms of shape.
Okay, so this goes beyond small worlds connectivity and things like that.
Yeah. It's, small worlds is just simply a statement about like the type of connectivity. So like, small worlds and scale free, those all basically have something to say about how the nodes are connected to one another in the aggregate. It's a statistical signature. You get it out of a single parameter. With other types of approaches, they're much broader, and people say, well, you can say a lot about a network by using like a network parameter, but what about like if you have a network that isn't a standard network? What if you have something that has high spatial structure, or something that is has information in the edges rather than the nodes that are it's more interesting. So none of those things are covered under the typical network parameter space. So you would go to some other type of method that would give you that information.
Okay, thanks.
Yeah. So you've got Vrutik, Ujjwal, Mainak, Shruti, and Abhishek are here today. Thank you. Hello. Oh, dem bones or boneheads.
You might explain that to people who don't know dem bones.
Oh, it's a song, and about kind of things that are connected to one another. So okay, so this is the satellite, and we have a couple of things here. Well, we have a number of different talks, a number of different sessions. The one we're in is the many and diverse signals of higher order interactions. There are also sessions, the spectral theory of hypergraphs, synchronization dynamics, and improved synchronization and forced diffusion in networks. So they're looking at a lot of different things going on in these networks. So that's that. And if you want to register for the networks conference, I mean, it's probably under $100 for students. I think well under $100 for students. So it's it might be doable, and they'll probably have the stuff online afterwards. So it's not you'll be able to participate in it at some level. So that's what I had on that. Let me go back to the submissions document to kind of tie up here. So we have those, and then the Google Summer Code, of course, we'll hear about that today. The Bacillaria non-neuronal cognition stuff. I received some emails between Dick and Thomas Harbeck, who's not Thomas Portegys, on this, and Thomas had some new microscopy movies that he captured from his cultures. He's still having a bit of trouble with the cultures, but he's collected some more data. So
you note what's the status of segmenting Bacillaria images now?
Well, I mean, we've had some people do some work on some of the projects. We had I think 7 people apply to the project, and so people have tried various techniques and need to catalog those and see what they are. But so we have a number of different techniques that are candidates for doing this.
Okay, I suggested that Thomas look at the question of whether or not Bacillaria motion is jerky, the way we found for single diatoms.
Yeah.
And his camera will go to 70 frames per second. Jerkiness was clearly observed at 10 frames per second, so he should be able to image it. I think he could use somebody to work with him in terms of segmenting those images and looking for whether the motion is smooth or jerky.
Yeah. So that would require a time series or a temporal analysis.
Yeah, even the stuff that's online is at 30 frames per second. It might be analyzable for jerkiness.
Yeah.
No. At any rate, perhaps you want to add that as a potential paper with Thomas. No. Our best is Bacillaria motion, smooth or jerky, something like that.
Yeah, I think I have that on the list. Let's see.
I don't doubt it.
Right here.
This came out of this weekend's conversation.
Okay. Well, let's
See,
see.
if it's smooth, it implies that something strange is happening, and it may be fundamental in terms of understanding the mechanism in general. Because there's no doubt that individual diatoms that have jerky motion.
Yeah. And then we'll make a note here that work with. All right. Yeah, that's. I'll put that on the list then. There's no deadline for that necessarily, but yeah, just collecting data and seeing from the data we have if we can get something. So, yeah, this is great, and Jesse's really interested in it. So we have a bunch of different methods that people have used, and so what would be required for this, if you're interested in doing this, if you've done a project, you submitted a project for GSOC on this, is to take images, and it would be over, you'd have like a time series of data, and you'd have to find sort of the derivatives of position. So it's like a higher order derivative of position. So each of your images is a certain position, and then that position you'll have to sort of find, there's some derivative where you average over, different frames and find like you're finding like acceleration, you're finding motion, and then, eventually you get to jerkiness, which is a derivative of sort of momentum, I guess. And so you're looking at higher order effects in the motion of these things. So like, we now understand the motion as a sinusoid, but there are different variations in that motion. And so that's really what we're trying to get at. Or some of those variations within the sinusoid, or, the sinusoid might slow down and speed up. And you've seen the movies, I think, a lot of you, where you're getting these kind of changes over time in the acceleration. So that's something that you might be able to do, and I don't know what technique would be optimal for that. But if anyone's interested, let me know, and we can work on that. So yeah, I put that on as number 26. I had number 24, which is molecular level simulations, but that's not really the same thing. So yeah, we have. I guess we actually had the NeurIPS. The deadline for abstracts is this week, and May 26th is the full paper. So I believe that Ujjwal said that he had some. He was working on a paper with someone on this in the group.
Hello.
Yeah.
Hello.
Yeah.
Hello.
In here. Yeah.
Yeah, so I've been last 2 weeks are very busy for me, so I haven't looked much into it. But for the last 2-3 days, I was looking and preparing a draft. So I think the abstract submission for the NeurIPS is difficult. Like it is deadline is today, so I don't know how much we can. I don't think so we can make it. But as far as the 26th deadline of full paper submission is depends. So I will talk to I haven't talked yet with the people I'm thinking to be working with, so I will definitely talk with them over Slack, and I will let you know. I have started preparing the draft as of now.
Yeah, that's good. Yeah, if you can't make the main NeurIPS deadline, there are a lot of satellite sessions that you can do that are more targeted towards like specific topics. The main track is very competitive. So, if you can't make that, then yeah, it's not like the end of the world. And definitely, though, it would be good to, it's always good to have a draft together. So.
So if we don't get into a NeurIPS, we can always try for some other conference as well. Like it will be good if we have work at least.
Yeah.
In my opinion.
Yeah, I think that's good. So yeah, there's that, and then we have the Mathematics of DevoWorm, which is still going. The Living Machines Conference, this is May 30th deadline. I think this is something I talked to Tom Portegys about. I know he's doing work on like his morphonostic stuff.
Okay, what was that? Friday?
Oh, so that was May 30th.
Oh, okay. I'll have to go check that.
Okay.
Did you ping me again about that? Because I can submit it.
Yeah.
Okay.
And here's the...
oh, yeah, now I remember.
Yeah.
Yeah. Yeah, okay.
I'm going to put the list... I mean, this is the document I'm looking at in the chat. So it'll be in, like, number 21 in that list.
Okay. Thank you.
You're welcome. So, yeah, that's something that is coming up this summer. And then, you know, we have these things that got rejected, a couple things. Well, one thing on the list here is the ANNs, BNNs paper. That was with Krishna. And then this is actually, I was looking at this submission, this one, Growth Form and the Theory of Deep Learning, which is the NetNeuro acceptance was, the name was a little bit different, but it's basically the same thing. I was looking at the connections between this submission and this ANNs, BNNs paper. So that paper, we worked on it together, like, you know, for this specific submission, and it didn't get accepted. But I think there's a lot of interesting stuff in there that can sort of, these two submissions can feed off of one another, 5 and 18. So, you know, we'll see where that goes. It'll be an interesting direction. Let's see. Then we have two other things on the list. The Boring Billion, which is this potential book contribution, and this is about, you know, early life and associated things. And then this Amazon Kindle book, which was another thing that we were talking about doing with the DevoWorm ML material. So DevoWorm ML was something we were working on in 2019. It was a set of lectures on machine learning and biology and developmental biology. So that might be something to put together. I started revising those lectures earlier, maybe like in January of this year, but I kind of lost track of that. I mean, I didn't really have time to do the other ones. So that's a chance to revisit that stuff and maybe make it more public. So, you know, we can put other things on the list too as we find them and put them on. Any comments about that? Okay. I know we had a bunch of people join the meeting after I asked this, but does anyone have anything they want to present today? Okay. Let's see. So if we don't have if no one wants to present anything, let's see. I actually, maybe I'll go right into the papers and do that. Let's see. All right, there we go. Okay. So there's a lot of stuff that's come out in the recent past, and the papers I'm going to present today are a bit, you know, oftentimes they're very machine learning oriented. Sometimes they're very biologically oriented. Sometimes I get into the biological papers and I don't know how much how much background people have in certain things. So it's always a challenge to know, like, what their threshold for tolerance is on some of this stuff. But we'll see where it goes. So a lot of the submissions I'm going to talk about today papers I'm going to talk about today are pretty biologically oriented. So the first one I'm going to talk about is this is so we were talking earlier in the meeting about non-neuronal cognition and single cell cognition and that sort of model of explanation for this stuff. And a couple weeks ago we talked about the Paramecium being a model organism for this. And so that was a different paper than this. The first paper we talked about was an eLife, and it was a paper, it was largely on the contributions of someone who has been somewhat forgotten in the history of this area, and their contributions and some of the experiments they did. This is a different paper, and this is a review of this area. So this is quite a healthy area, I guess, of inquiry. This is integrative neuroscience of paramecium, a swimming neuron. So again, paramecium doesn't really have a nervous system per se, not like you would find in C. elegans, say, with the connectome. But this is a paramecium, which is a single-cell organism, but nevertheless, it's called a swimming neuron. And so, let's see. So the abstract is, Paramecium is a unicellular organism that swims in fresh water by beating thousands of cilia. So cilia are these little, well, I call them cilia, but that would be not really descriptive. They're long, things that move back and forth. I think, like a brush, it has a lot of bristles that are, flexible, that move back and forth. And they move them against the water current to move around. When it is stimulated mechanically, chemically, optically, thermally, so just a number of different modes of stimulation, it often swims backward, then turns and swims forward again. So this is a single-cell organism that has sort of a set of behavioral modes that has like a set of appendages that move it around the environment. So it has this sort of setup where you would think, well, it needs a nervous system, but it, of course, being a single cell, it really can't. So how does it do this behavior? And so you're basically stimulating the single cell in different modes, and then it interprets that signal as, swimming backwards, and then turning, then swimming forward. So you actually see this behavior in C. elegans. If you look at our data for the, we have a lot of movement data, and a lot of the movement data is sort of, it describes certain movements. So, you can see all sorts of stereotypical movements in C. elegans. Movement behaviors with respect to like poking it with a little pick, or putting food in a certain location, and they'll explore for food, and they'll make these different what they call omega turns and other types of movements. They'll move backwards and then forwards again, and so they're doing this. But C. elegans has a neural circuit for this, and the neural circuit is actually well studied. And if you look at some of the papers that have been published on like feeding circuits or like chemosensory circuits, and Tom actually has worked on this in terms of making this into a robot, you can see that there's a certain pattern in that circuit, but that requires a number of neurons that are connected. In paramecium, you see these things, but it doesn't involve a nervous system. So this avoiding reaction, which is a simple avoidance reaction, is triggered by a calcium-based action potential. So it's actually an action potential like you see in neurons, but it's not in a neuron. It's actually in a cell. And so we've talked about Michael Levin's work, where he's looked at the electrical activity of cells that aren't neurons. So, for example, somatic cells of different types, and they have maybe not action potentials, but they have electrical potential. In this case, you have a calcium-based action potential, or at least what they claim is a calcium-based action potential. That is an electrical current in the cell that is exploited for this purpose. For this reason, some authors have called paramecium a swimming neuron. This review summarizes current knowledge about the physiological basis of behavior in paramecium. So I think the last paper we talked about, paramecium, talked about the ability maybe of paramecium to learn in an associative manner, so associative learning. So this is something we associate with like a simple neural network, maybe like 2 neurons or 3 neurons in a circuit. And, like there's this co-stimulation that goes on, and it can associate one thing with another in the environment. So in the, we talked about the sea slugoplegia, for example, they exhibit this very simple associative learning. And so you can see that in a simple nervous system. You can see it even more so in larger nervous systems, but you even see it in a single cell here. So this paper it's a review. It kind of goes through what paramecia look like. So this is the paramecium. These are the cilia here. So they look like little hairs, and they move, but they move in a way that's a response to the environment. So if it gets stimulated, it moves in a certain direction, and it can actually exhibit these movements where it moves backwards and then forwards. This is the anatomy that, from 1899. They drew this out. Back then, they used to draw things out really in a detailed manner. Now they just kind of take high-resolution pictures. So it's kind of a lost art in biology, is all these kind of drawings that people used to do. So, yeah, this is 100 to 300 micrometers long. So this is something that, it's about, actually, it's maybe a little bit bigger than your average diatom. So these aren't very big, these organisms. And they've been around for millions of years. And it's a somewhat popular model organism. So, and this is the avoiding reaction here. So you can see that there's this, so A here, avoiding reaction against an obstacle is illustrated by Jennings. So this is the obstacle here. A, this circle, the paramecium encounters this obstacle. So when it does this, it moves backwards. So it's receiving pressure stimulation to the front of the organism or the cell. It starts moving backwards, and then it makes a turn and it moves forwards in this direction, ostensibly to avoid this barrier or this obstacle. That's basically the movement we're talking about. And in response to this movement, you see an action potential here in response to a 2 millisecond current pulse, which is, there's a whole field of action potential analysis, so I don't want to get into that, but basically it's this rapid change in potential within the cell membrane. So you see this change in voltage. And so they talk about the life of a paramecium, swimming, feeding, and reproducing. So this is a mode of movement they call spiral swimming, where there's this is one of their modes of movement. B is thigmotactic paramecium resting against a fiber. I can't remember what a thigmotaxy is. I think it's like a water pressure or something like that. So, and then you have, so it's a taxi is a movement in response to some stimulus. So I think we've talked about taxis before, and this is one example of this. And then two paramecium in conjugation, sexual reproduction. So they actually sexually reproduce, and they have movements associated with that. And so they also can navigate. So they do this backing up and moving in a different direction. So that's well characterized. So this is, if we're talking about non-neuronal cognition, this is one example of how this works in single cells. And of course, we don't know if this is something that is goes beyond paramecium, but it's definitely, we have a good model for understanding sort of the basis of it. What are the important things to look at within the cell? So you also see chemotaxis and social behavior. You see a bunch of paramecium gathered around the circle. It's a drop of weakly acid solution, so they drop some acid into this area here. They aggregate around a drop of weakly acidic solution here. And then in this case, C, they actually are avoiding a drop of sodium carbonate. So there's a drop of sodium carbonate in the culture, and they disperse. And they actually avoid this spot. So this is a social behavior. I'm not really sure how much they communicate with one another, but it looks like a collective behavior. So, and then they can also gather in like a cloud of carbon dioxide generated by their respiration. So they can take on this phase of clustering. So all these are social behaviors, but we don't know, when they say social behavior, they don't really talk to the paramecia. It's just kind of, it looks like collective behavior. So this is that paper. That's let me make, I'll put a link to the paper in the chat here. But you can go through the rest of the paper, and it really kind of gets into a lot of detail about a lot of the different behaviors. And they have a very diverse set of behaviors that they exhibit. So definitely, if you're interested in non-neuronal cognition, it's something to look into. So let me put the link to the papers in the chat here. And then let me see what we have with respect to our chat here. If you only call them cilia, that would be silly, yeah. Yeah, it would be. I was going to make that pun. I didn't go there, but Jesse did, so thank you. Yeah, so that was his reaction to that. Okay, so then that's that paper. The next one is, what is this one? This is an image that I found. Oh, this is kind of an interesting talk that I didn't see the talk, but this is just something to be aware of here. This person gave a talk on floral cell type diversity. Reminds animal biologists how beautiful plants are, and almost makes you want to do serial EM on a flower. So flowers can be very diverse and make for some very good subjects for imaging studies. So this would be, this is they do a lot of. We don't talk much about plants in the group. I just wanted to make the point that plants are still there, and they're very much, you can observe, development in plants. You can look at their embryos, and you can also look at flowering plants in terms of like anatomy and other things. So the next paper, this is going to be a tough one, I think, to get through. But so this is a paper on what they call syntitia, or syntitial blastoderm formation in a cricket. So a cricket is a cricket that is, it's an insect. Syntitial is a word, I don't know if people know what that means. It's basically it's a sort of a cell mass with a bunch of nuclei in it. So often in C. elegans, we know that like muscle forms from a synticium. So syntitium is a tissue that has a lot of nuclei in it, and then it diversifies into muscle. And so this is a type of thing. They're looking at nuclear movements during this blastoderm stage, or blastoderm formation stage. And so I think we've talked about Cassandra Extraver before, her work. This is one of she's on this paper. So the abstract reads, animal embryos pass through an early stage called the blastoderm, in which cells are arranged in a continuous layer at the periphery of the embryo. And so this is where they're sort of, I guess it's kind of like the ring that I showed you before, where you have like there's an empty space in the middle, and then there's a ring around the edge of the embryo of cells. And this is the blastoderm. Despite the broad evolutionary conservation of this embryonic stage, the cellular behaviors that led to blastoderm formation vary across animals, and the mechanisms that regulate these behaviors are poorly understood. In most insects, pre-blastoderm development begins as a synticium, that is, many nuclei divide and move throughout the single shared cytoplasm of the embryo. So this is where, again, you have this development begins as a synticium in insects. So you have a lot of nuclei in a shared cytoplasm. So it's like a single cell. It's not really a single cell, but and you'll see this if you look at some of the data we have on Drosophila, where they have like this huge cytoplasm with a lot of nuclei, and then there's a cellularization that happens where you just get independent cells, so that cytoplasm sort of partitions into different cells. Then these syntitial nuclei must move from their scattered positions within the cytoplasm to form a single layer at the cortex. So these nuclei basically move out into a layer. And so the nuclei, of course, is where the DNA is, and the thing that we look at usually when we look at microscopy of like cells, we'll often put like a fluorescent marker in the nucleus, and that's how we can mark a cell. It's one way to mark a cell, and so people use this. It's not really the center of the cell, but it's a useful point of reference when you're looking at cells, especially when they're migrating. Then these syntitial nuclei must move from their. Okay, recent work has shown that in the fruit fly Drosophila melanogaster, Some of these early nuclear movements are caused by pulses of cytoplasmic flows that are coupled to synchronous divisions. So this is, like different types of waves and flows in the cytoplasm early on. We've talked about different waves of calcium waves and things like that, differentiation waves. And then they're coupled to synchronous divisions. Here we show that the cricket, Gryllus bimaculatus, has an altogether different solution to the problem. So in Drosophila, they do one. There you can observe one type of thing. In this cricket species, you observe something very different. We quantify nuclear dynamics during the period of synticial cleavages and movements that lead to blastoderm formation. So these synticial structures, they kind of, they partition themselves into cells, and that's what they call the cleavages. And movements that lead to blastoderm formation in the embryos with transgenically labeled nuclei, which are these fluorescent labels that we often see in these in the microscopy images. So when you see a black background with a lot of light, points of light, those points of light are these nuclei that are labeled. And they use something called a GFP or an RFP or some other color of fluorescent marker to do that labeling. So that's what, when you run across any of that terminology, that's what that means. We found that, one, cytoplasmic flows were unimportant for nuclear movement. So these flows were unimportant for the nucleus moving around. Didn't really coordinate the movement of the nuclei. And two, division cycles, nuclear speeds, and the directions of nuclear movement were not synchronized across the embryo, as in D. melanogaster, but instead were heterogeneous in space and time. So they weren't coordinated like you see in Drosophila. They were kind of, I don't know if they were random, but they were definitely heterogeneous. Moreover, several aspects of nuclear divisions and movements were correlated with local nuclear density. We show the previously proposed models for the movement of D. melanogaster synthetical nuclei cannot explain the behaviors of the nuclei in the cricket. We introduce a novel geometric model based on asymmetric local pulling forces on nuclei, which recapitulates the density-dependent nuclear speeds and orientations, without invoking common paradigms of localized polarity cues or cell lineages determinants of nuclear activity. So our model accurately predicts nuclear behavior in embryos. We show the model can be used to generate falsifiable predictions about the dynamics of blastoderm formation in other species. So this is they kind of go through this here. They talk about the comparison with Drosophila. And there's some methods. I want to get to the images. They're in here. Usually they're at the end. Okay, I think they're here. So this is the first image here, and this will make this a lot clearer what I just said. So this is an example of a time lapse here. So this is the synthetical blastoderm. So you can see this whole thing is basically like open cytoplasm, and you have these nuclei that are stained. So these black dots are the nuclei, and you have this time lapse where you can see 150 minutes since the start of the time lapse, 300 minutes, 450 minutes, and then 24 hours. So you start off with a few identifiable nuclei here, then you get more nuclei scattered about, even more, and then you get this sort of, they're all over the synticium, and then you get this, and as this is happening, it's partitioning this cytoplasm. And then you get the cellularization and embryonic rudiment formation. So now you get this cellularization happening at at this time point here, and then at 24 hours, this what they call the embryonic rudiment is here. And so you can see that there's this is how they sort of move about in the synticium, and then end up forming these cells. And so they have this, actually they show the nucleus here, and they show some segmentation of the images so that they're looking at the nucleus, and then they call the energid, and then the cytoplasm. So they can actually do that during development. And then this B here is nuclei retract to produce a 3D plus T data set of nuclear lineages. So here they visualize this as a set of nuclear lineages. Instead of cell lineages, which is something we're very familiar with in C. elegans, there's a nuclear lineages to look at how these things divide and actually move around the synticium, which will end up being a bunch of cells. And so all nuclear tracks are displayed for an example embryo with the lineage descended from a single nucleus highlighted. So I think this is the starting point here, and then it kind of they trace the lineage across like this. So that's that figure, and then this figure actually shows, let's see, figure 2C is a cell cycle duration was positively associated with local density. So cell cycle duration was calculated in the vicinity of each nucleus by measuring the time elapsed until the number of nuclei within a 150 micron or micrometer radius increased by 25%. Top row shows 3 example time points with local cell cycle duration displayed as colored volumes, each of which contain a single nucleus. So then this is the evolution of this over time. So at 300 minutes you have this cell cycle duration, it's sort of heterogeneous here, and then it really gets heterogeneous at 413 minutes. Cell cycle in different patches here are much longer than in other places. And so then this is actually, figure 3 actually shows these trees that they're generating from this. So this is the lineage, the nuclear lineages that they have. So they have the nuclear position, anterior being the front, posterior being the back end of this structure. The nuclear positions, of course, are spread out across this. They start off, they start measuring a couple of them, as you saw in the first figure, and then they start off in a couple of positions, and they start to divide, and you start getting more and more, and they spread out across the synticium, and you can see the pattern here where they're dividing and they're moving, and you see that they end up populating the entire length of the embryo. And so this is the pattern. And they actually have a speed diagram here where they show the speed of migration over time. So this is all good stuff. And then this is an example of how they've taken these nuclei and they've done these Voronoi models. And a Voronoi model is where you take a bunch of points and you try to draw boundaries around, you try to find the sort of the midpoint between these points and draw a boundary. So these are supposed to be like optimal tilings. They sort of describe these points as sort of centroids in a geometric model. So you draw boundaries at roughly the midpoint between the two most adjacent points, and you end up with this set of boundaries which sort of define maybe what the cells will look like as they emerge from the synticium. So this is a Voronoi decomposition that they used here. So I don't know how many of you I think we've talked about this many years ago in this group, but this is a method you can use to sort of segment, it's a segmentation technique, but you don't use it for every type of segmentation. It's very specific to, sort of what you might expect the optimal outcome to be. So there are a lot of nice figures in this paper. So I think if you're interested in this area, this is definitely a paper to check. It's definitely a different model system than we've seen. I'm not really sure what I know that crickets aren't really, as far as I know, they're not really a very popular model system. But it's definitely a nice, you can find out different information by looking at systems, different systems. So, yeah, there are a lot of really nice pictures in here. It's really great. So that's that paper. Then we have, let's see, this paper here. I think this is something that Susan sent me a long time ago. This is, and I hadn't gotten to it yet. This is called 3D Viscoelastic Drag Forces Drive Changes to Cell Shapes During Organogenesis in the Zebrafish Embryo. So this is a zebrafish paper. This is about viscoelastic forces, which are, we've talked about drag forces and things in embryogenesis, that they have an effect on position and migration. And so this actually drives changes to cell shape during organogenesis. So the highlights of this paper are that new physics-based simulation methods allow study of dynamic tissue structures in 3D. Movement of an organ through tissue generates viscoelastic drag forces on the organ. These drag forces can generate precisely the cell shape changes seen in experiment. And PIV analysis, I don't know what that is. Well, I guess we'll find out. They're experimental data matches simulations. So for experimental data, they'll match the simulations, and it allows you to probe tissue mechanics. So these are the PIV, and this is the PIV analysis here. And then this is the simulation that they do on the same thing, and these results match. I don't know, Susan, did you want to talk about this paper at all, or?
I'm not exactly ready to talk about it because I've been writing a 6,000 word essay, but yeah, there's some interesting movements here. There's a circular movement, and then the whole circular movement tends to move up the embryo. I think up, I don't know what you would call that, but up or down, which way? It looks like up from the arrows. Anyway, the cells ahead of it become calmer, and the cells behind flatten out, so that provides the driving force for the circular swirling bunch of cells, and they create a shear, and they cause left-right asymmetry when that occurs. So it's all a part of how a vertebrate, in this case a zebrafish, develops a backbone.
Okay.
So I think it's really interesting.
Yeah, that's great, and that's pretty important part of embryogenesis. Thanks for the comments. Yeah, yes. Hopefully your exam is going well there. The essay that you're writing.
It's getting too long, I would say.
Okay. We had a couple of comments here. Synthesia, C is pronounced as an S. Synthesia. Yeah. So Tom says, don't slime molds have multiple nuclei in a shared cytoplasm? I think so. I'm not really sure, but that's maybe true. Yeah. And then Aswarth says, hello everyone, my name is Aswarth and I am a CSE undergrad from India. I looked into the repo of DevoLearns. I tried to solve one of the issues there. I attended one of the previous meetings. He's interested in the cellular automata work that Shruti is doing. Fascinated him a lot, made me feel motivated about contributing to the community. I'm not able to understand where to start. So, Aswarth, welcome. And yeah. And so, yeah, we can talk, like, are you part of the Slack?
Yeah, I was I joined it, but I was not active. Now I'm in a better position to be active.
Okay. Well, I mean, we often, if you join the DevoLearn channel, which is not the DevoWorm channel, but there's another channel, DevoLearn. There's sometimes issues are posted there, and just keep checking out the DevoLearn channel or the DevoWorm channel as well, and you can maybe ask around. You can inquire about things if people want to talk about certain issues that they're interested in. And then Shruti, who's in the meeting, she's been presenting on the cellular automata, on neurocellular automata. So she's working on this thing with respect to that, this project. So you might want to talk to her about maybe some of the things that she's been presenting on.
Yeah, that I will look into.
Okay. Mainak said PIV is particle image velocimetry. Yes. PIV analysis is used to measure flows of planar velocity fields. Yeah, that was the they had this right here. Yeah, so that was what they were doing in this paper, that type of analysis. Dick says slime molds, yes, some are 100 meters long in seaweeds, and this is with respect to slime. Yeah. And then Krishna is not here because his father is in the hospital unconscious with a stroke. Well, sorry to hear that, Krishna. I hope your father is feeling better soon, and hope you're doing well. Yeah, sorry to hear that. So yeah, that's our chat. So yeah, that was the paper, and then I think I'll do one more paper before we go. And this is kind of a shift from what we've been talking about here. It's back to sort of humans and mammals, and I'm not going to give this paper what maybe the attention it deserves, but this is, I thought I'd talk about it just to, just let it wash over you again. This is not like, you're not going to have an exam on this or anything. And this is the cellular and molecular landscapes of the developing human central nervous system. So this is a series of neuroscientists who are interested in the development of the nervous system. And so the human CNS follows a pattern of development typical of all mammals, but certain neurodevelopmental features are highly derived, which means that they're, through evolution, they've made a lot of changes from sort of the common ancestor. Building this human CNS requires the precise orchestration and coordination of myriad molecular and cellular processes across a staggering array of cell types over a long period of time. It's like human development, of course, is we're in the womb for 9 months, and then there's a lengthy developmental period outside the womb, going from maybe like, a year to 7 years, maybe even to over 20 years for some things in the nervous system. So if you ever take a course on, sort of neurobiology or developmental neurobiology, you'll learn that, in the first year our synapses are pruned. We're born with a much different brain than what we had, what we end up with at a year after birth. And then after about 7 or 8 years, there are a lot of neurochemical changes in the brain. And then in puberty, there are a lot more neurochemical changes in the brain. And our brains don't truly mature until we're in our 20s. And then people have questioned whether this has something to do with plasticity. You still observe plasticity even in adults. So this is a very long process relative to the organism. Dysregulation of these processes affects the structure and function of the CNS and can lead to neurological or psychiatric disorders. Recent technological advances and increased focus on human neurodevelopment have enabled a more comprehensive characterization of the human CNS and its development in both health and disease. The aim of this review is to highlight recent advances in our understanding of the molecular and cellular landscapes of the developing human central nervous system, with a focus on the cerebral neocortex, which is the sort of the top of the brain, what we used to think with, like a lot of cognition happens there, and the insights these findings provide into human neural evolution, function, and dysfunction. So that's a lot that they're going to set out to do here. So this is really, this is, again, a review, so you have a lot of citations in the front, in the introduction, and they talk about the prolonged developmental course and period of dependency. So, we're more than other primates, we need to be in a parental setting for 18 years or, 10, depending on your culture. People like maybe a couple hundred years ago didn't have the length of childhoods that we do today. But this is, this is all due to our very long developmental trajectory for our brains. And so we're dependent often on our parents for a long period of time. And in a lot of organisms, they're just kind of born, if they're not born live, if they're born from eggs, say, they're all ready to go when they hatch. So that's a huge difference in terms of like the biology of the brain. In C. elegans, for example, the eggs hatch and they go through these larval stages, but they don't need parental supervision in any way. in humans, you need a large amount of parental supervision. So, and a lot of that has to do with the development of the brain. And so in our central nervous system, we have things like proliferative zones and diverse subtypes of neural stem and progenitor cells, which actually help our brains. Our brains have expanded across evolution, so it helps with that, but it also helps with neuroplasticity and some of the things involved in that, like neural repair and other types of things. And there's a whole literature on neuroplasticity that I won't go into. The other group that I had, we talk about that actually much more. So if you want to find out more, let me know, and maybe we can get into that, get in touch with that group. So the emergence of many of these neurodevelopmental processes that have enabled the complexity of the human brain may have required a trade-off that rendered it particularly susceptible to certain disorders. So that's interesting, that a lot of disorders in the brain may be due to the complexity of the brain. And so we know that from model organisms we can study things about the human brain. People get a lot of funding to study, say, for example, different different diseases and other things in other organisms. So, for example, C. elegans is used to study aging, and there are other organisms that are used to study addiction, like mice. And, there are other organisms that are maybe very phylogenetically distant from humans that are used to study diseases. So a lot of these diseases have sort of roots in the genome that you can, use like phylogenetically distant organisms to understand, but some of these things are really kind of unique to the complexity of the human brain. So, and then this is a nice figure. This is actually a really big figure. This is a timeline of key human neurodevelopmental processes and functional milestones. So this is like sort of if you're interested in neural development in humans, this is the figure right here. They have the sort of the time series of different things in the brain as it's developing. So this is in the embryo, in the fetus. This is where you get, this isn't fetal development. This is sort of like embryonic development. Then this is fetal development, these three brains. So you get these structures, you get a neural tube, and then you get elongation, and you get these different structures in the embryo. And then in fetal development, you get these other structures that start kind of looking like a human brain, or at least a mammalian brain. And then at birth, you start to get these bigger brains, and you start to get what they call convolutions on the outside of the brain that are forming in, as you're heading towards sort of in between this birth and newborn stage. So it's very early, and then you're starting to get, this is like 3 years old, 30 years old, and 90 years old. And over time, the brain after about 30 years, the brain starts to decay, at least age enough so that it loses cells, it loses connections. But, that's something that can be overcome by exercise and other things. There's a whole literature on this, like I said, about neuroplasticity. And they actually break this down by region and function and mechanism. So myelination, synaptic pruning, synaptogenesis, they have these all labeled on this timeline. So, and then also, functional milestones, so different types of, sensory motor stimulation, communication, cognitive achievements, they're all on this timeline. So this is largely, focused on development up to the newborn stage or up to like the first year of life. And so this is a handy sort of figure for that period of time. And then they show, of course, the onset of different disorders. So they try to link this to development, and not only development, but what they call life history, which is this period of, how does the organism how does the organism's trajectory play out over the course of life? So, you have development, but you also have life history. So certain things happen in your youth, certain things happen in your middle age that affect your later life. There are other processes that unfold during the lifespan that are, that are actually evolutionarily selected for and things like that. So there are a lot of things going on with life history as well. And then they have this human CNS in numbers, which is where they give a lot of numbers. And these are like cell types and number of cells. So these are kind of estimates that people have come up with. So this is why they sort of have all these citations in here. The number of neurons in the human brain varies by as much as a factor of 2. So we don't know how many cells there are. Like in the C. elegans connectome, we know exactly how many cells there are. And it's always the same number of cells across every C. elegans unless it's a mutant. In the human brain, we have no idea how many cells there are. Furthermore, there could be a lot of variation, individual differences, or, by age. So you can have, but it's immaterial because there's so many neurons that, it's not it may or may not have any sort of functional impact. So this goes on for quite a while. And if you're interested in this topic, you should definitely check this out. They talk about something called the transcriptional landscape, which is where they look at gene expression, and these are the figures that they use to look at that. So they usually get like a bunch of data on gene expression, and then they look at the data, and they use some sort of dimensionality reduction to make these maps, where basically if you have these clusters, and we've talked about dimensionality reduction, but if you have these clusters, that tells you something about the data. So that's that paper, and a lot of references in here. So if you're interested in that, I don't know if I posted the paper list in the chat, but let me post it again so that everyone has this. And again, if the sharing is off, let me know. I can put the thing. So Susan said she has to leave now. The farmers here need a ride. Thank you for attending, Susan. Jesse says, can you link to the paper here? Okay, this is the link here. I just posted the folder, so if you're interested in that. We have, Dick said, tried PIV once on a diatom, but PIV software couldn't handle the diatom's motion. And maybe now they actually have better software for it. I don't know if this was a long time ago or what, but and then number of cell types in brain speculated to be number in body, and Gordon, the hierarchical differentiation or genome and differentiation waves. Yeah, so there are a lot of estimates about number of cell types. There's that book Bonner's book. John Bonner published a book in 1988 about speculating on the number of cell types in different animals. But then there are people who have tried to do a lot of microscopy and machine learning to try to differentiate the number of cells using a number of different criteria.
You missed the greater than sign.
What was that?
You missed the greater than sign.
Oh, yeah, greater than in the body, yeah. Yeah. So, I mean, it's like you can use a number of different criterion to know how many cell types there are. Like you can use morphological criterion, you can use molecular or genome criterion, so you can look at like what's being expressed in the cell, and if it's different, whatever that means, then it's a different cell type, which is fraught with a lot of problems, because you can imagine that you have to know why something is upregulated or why something is different. Maybe that's just natural variation in a single cell type, or maybe it's two different cell types.
Yeah, I recall a lecture from the 1960s when RNA was just discovered, that there was more kinds of RNA in the brain than anywhere else.
Yeah. And it's because of that need for functional diversity. Things need to be, turned on and off as by, given in a given context. A lot of circuits are reused for different things. So, and then, it's, there's this idea of lumping and splitting when we talk about categories. And that is like, if you want to classify things in the world, do you want to put them in a small number of categories? Is your criterion rather tight or rather loose? Like you say, basically, if it has this set of properties, maybe a small list of properties, it's one of maybe 5 categories. Or do you want to split infinitely into many, many different categories, and you want to use like this very strict criterion for each of your categories. And we talk a lot about machine learning in the group, and one of the things that we don't talk about too much is lumping and splitting, which is where you either have like a loose criterion for categories and you have maybe a few broad categories, or you have these infinitely this infinite number of categories, and it's just really anything that's different is going to put you into a new category. And so this has been an issue for hundreds of years, actually, in systematics, biological systematics, and in other types of classification, where really you have to make a decision ahead of time as to what your categories look like and what's important to you in terms of understanding your data. So if you have a lot of potential categories that you want to know, like you want to, then you would use a lot of criterion, and you would, and both approaches are useful in the sense that, you may not want 1,000 categories. You may only want to know about a couple of certain key elements to your data. On the other hand, if you have 1,000 things you want to know about, then splitting is better. And Dick says, does anyone here have access to PIV? I don't think so. Sure. I'm not sure what, yeah. Well, anyways, if you can answer Dick's question, if you have access to it, or the state-of-the-art software for this, let us know. So, okay, so thanks for attending. I don't know if anyone has anything else they want to say before we wrap up for today. Does anyone else have any comments or questions?
Good luck to everybody, and I think next week with the GSOC Cup coming out and some other things, I look forward to kind of making the plan ahead. So I'll be playing more than actually, particularly in June. When June comes, I'll be a little bit more active then. Hope everybody's doing well, and hope Christian family does well too.
Yeah. Yeah, I hope Christian is doing well. Mainak says have a great week ahead, so thank you. And again, if you, thank you. Take care. And if you don't get selected for GSOC, or don't be disheartened, it's just very competitive, especially last year, and I take it this year as well. So we shall see. Thanks for attending. I know that Dick and, bye, Tom. I know that Dick and Shruti, I think, are going to meet now, so I'm going to leave. And if you want to stick around, do so. Otherwise, have a good week. Thanks.